Assalamualaikum and hi to everyone. Today we are going to learn chapter 9 Homeostasis, subtopic 9.1 Concept of Homeostasis and 9.3 Human Homeostatic Organ, Structure and Function of Kidney. This chapter will cover 3 marks in UPS 3 and 6 marks during PSPM. Homeostasis is a process of maintaining biologically constant condition inside a living organism through physiological processes. The term biologically refers to, for example, physical factors such as blood pressure and body temperature, or chemical factors such as blood glucose level and pH. The importance of homeostasis: number 1, enable adaptation of organisms to the changes of external environment. Number 2, constant internal environment enable the chemical reactions to occur in the cells. And number 3, enable organisms to live in a different habitat with a wider range of environmental conditions. Homeostatic mechanism involve regulatory mechanism which based on negative feedback mechanism. The homeostatic control system involve the 3 components. Number 1, receptor. Number 2, control center. And number 3, effector. The first component is receptor. Receptor will detect a particular stimulus of changes in physical or chemical factors and send impulse to the control center, which is central nervous system. For example of receptor is chemoreceptor or thermoreceptor. Second component is control center. Control center will receive and translate impulse from receptor, for example, control system or CNS, and send impulse to the effector. The third component is effector. Effector will receive impulse from control center and produce response or corrective mechanism action through negative feedback mechanism to restore condition back to the normal. An example of effector is muscle cells or glands. Kidney is an organ for osmoregulations and excretion. What is osmoregulation? The maintenance of correct balance between water and dissolved solids in blood or tissue fluid by controlling the volume of water, ion concentration, osmotic pressure and pH of blood or tissue fluid. Whereas excretion refers to the elimination of waste products of metabolism from body. Each kidney is supplied with oxygenated blood from aorta through renal artery, and renal vein carry the oxygenated blood from kidney through inferior vena cava. The cross-section of kidney consists of 3 regions, which is renal cortex, renal medulla, and renal pelvis. Renal pelvis is a large urine collecting space inside the kidney. The medulla region is the middle portion of kidney that consists of lobes of Henle and collecting duct. Cortex is the outermost region of kidney that consists of glomerulus and Bowman's capsule, convoluted tubule, including proximal and distal tubule. Nephron is the microscopic and functional unit of kidney. In each kidney, there are 1 until 1.5 million of nephron. Each nephron consists of vascular component and tubular component. There are 5 structures in vascular component. Number 1 is afferent arteriole that carry blood from renal artery to the glomerulus. Number 2 is glomerulus the network of capillary between afferent arteriole and efferent arteriole. Number 3 is efferent arteriole that carry blood away from glomerulus. Number 4 is peritubular capillary, a network of capillaries branch from efferent arteriole that surround the convoluted tubule. Number 5 is vasa recta, the peritubular capillaries that surround the lobes of Henle. And the last one is renal vein that carry blood away from kidney. Whereas for the tubular components of nephron, we have Bowman's capsule, proximal convoluted tubule, lobes of Henle, distal convoluted tubule, and the last part is collecting ducts. The Bowman's capsule is the structure that encloses the glomerulus. The outer layer consists of simple squamous epithelial, and the inner layer consists of the cell that we call podocytes. The Bowman's capsule involved in the ultrafiltration process. Podocyte is the cell that surround the glomerulus or glomerular capillary. It has many foot-like projections called pedicels or foot processes that attach to the basement membrane. Podocytes are not tightly arranged and leave slits between them in order to provide filtration slit. The filtration slit is covered by slit diaphragm to ensure that large micromolecules such as albumin and globulin will remain in bloodstream, whereas small molecules such as water, glucose, ionic salt are able to pass through the slits diaphragm, thus forming an ultrafiltrate which is further processed to become urine. Basement membrane. The layer lies between endothelial of glomerulus and podocytes. It functions as to prevent large molecules such as protein, blood cell from leaving the blood. The second structure of tubular components is proximal convoluted tubule. It consists of one cell-thick cuboidal epithelial cell, and the inner surface is lined with microvilli that increase the surface area for reabsorption, and the cuboidal cell contain mitochondria which provide energy for active transport. Proximal tubule involved in reabsorption of substance from tubule into the blood and secretion of substance from blood into the tubule. The next part of tubular component is lobes of Henle. It composed of two parts. The first is descending limb of the lobes of Henle, this one. Descending limb is lined with simple squamous epithelial cell and has numerous water channels that formed by aquaporin. This characteristic will give the permeability to water and no channel for salt and small solute. The second part of lobes of Henle is ascending limb, this one. It has two specialized regions. First, thin segment of ascending limb, this one, that is lined with simple squamous epithelial cell, and thick segment of ascending limb, this one, that is lined with cuboidal epithelial cell. Ascending limb lack water channel that give the impermeability to water but permeable to NaCl or salt. The lobes of Henle involve in the reabsorption of water or salt and thus enhance the urine concentration process. The second last part of tubular component is distal convoluted tubule. It composed of cuboidal epithelial cells, and the inner surface is lined with few microvilli and mitochondria compared to the proximal convoluted tubule. Distal convoluted tubule involved in reabsorption and secretion to regulate the potassium ion and NaCl concentration in body fluid, and to regulate the pH by control the secretion of hydrogen ion and bicarbonate ion. The last part of tubular components is collecting duct. The presence of water channels or aquaporine in the collecting duct epithelium is controlled by the hormones that regulate the blood pressure, volume, and osmolarity. Collecting duct involved in a reabsorption of solutes or urea or salt or water. Urine formation process involves the mechanism that we call countercurrent multiplier. Countercurrent means filtrate flow through loops of Henle in opposite direction, for example, down the descending limb and up through the ascending limb, whereas the term multiplier means the osmotic concentration of filtrate become progressively higher in descending limb and become progressively lower in ascending limb. Loops of Henle has two limbs. First, descending limb that is permeable to water but not permeable to NaCl, and ascending limb that is permeable to only NaCl but impermeable to water. In ascending limb, there are thick segments where NaCl is actively transported to medullary region, whereas in thin segment, NaCl diffuse out to medullary region. This happens due to the difference of NaCl concentration between filtrate and medullary region. In this mechanism, water diffuses out from descending limb into the medullary region by osmosis. This is due to the permeability of descending limb to water and result in production of concentrated filtrate. As we can see here, the number of filtrate start from 300 and become progressively higher until 1200 value, which is the most concentrated filtrate. NaCl diffuse out from thin segment of ascending limb, whereas NaCl is actively transported from the thick segment of ascending limb to the medullary region. This result, the filtrate become more dilute as it moves to the distal convoluted tubule. In the inner medullary region of kidney, the ducts become permeable to urea, and because of high concentration urea in filtrate, some urea will diffuse out of duct and move into the interstitial fluid. Along with NaCl, this urea contribute to the high osmolarity of the interstitial fluid in the medulla and result in the water will be diffused out of descending limb and also collecting duct into the medulla by osmosis. Finally, the reabsorbed water will diffuse into the vasa recta that surround the loops of Henle, and urine in the collecting duct will become more concentrated as it moves to the renal pelvis due to the reabsorption of water.